Hello everyone. Uh, in today's lecture we will continue talking about amino acids oxidation. In the part 1 in earlier lecture we talked how amino group from amino acid was removed through transamination reactions, and it was carried in form of glutamine from all the extrahepatic tissues to liver, and in the liver that ammonia was converted into urea through urea cycle. So far in the previous lecture we have seen that the intercellular proteins and also dietary proteins, they were hydrolyzed into amino acids, and in the first reaction of amino acid catabolism, amino group was removed in form of urea and carbon skeleton was left. This ammonia enters into urea cycle through carboxyl phosphate, and eventually urea is produced, that is excreted out from the body. So this we have covered in the part 1. In today's lecture we will discuss about fate of this carbon skeleton which is left after deamination reaction of amino acid. So this carbon skeleton is converted into alpha keto acids. Depending on the amino acid, alpha keto acids are produced, and those alpha keto acids, they enter into citric acid cycle in order to get converted into carbon dioxide and water, and to release ATP, to synthesize ATP in oxidative phosphorylation. And also oxaloacetate also produce in this reaction, the catabolic reactions, that oxaloacetate could enter into gluconeogenesis to produce glucose. So on the basis of the end product of the fate of carbon skeleton, amino acids can be categorized into glucogenic or ketogenic. Ketogenic amino acid, they give rise to acetyl coenzyme A, whereas glucogenic, their end product is used in synthesis of glucose. So in this lecture we will talk about oxidation of carbon skeleton of amino acids, all 20 amino acids, and we'll talk about ketogenic and glucogenic behavior of different amino acids. We also talk about branched chain amino acids and their role as bodybuilding supplements. Generally, the people who do a lot of exercise, spend a lot of time in gym, and who like to build muscles, they take branched chain amino acid as food supplement. So we'll talk about that, the physiological reactions, the biochemical reactions involved in oxidation of branched chain amino acids and their role in building muscles. Then we'll discuss about genetic defect due to amino acid metabolism. If some enzymes of amino acid catabolism are defective, they are mutant, in that case the disorder, metabolic disorders arises. So we'll discuss about those, and also we'll talk about defect in the urea cycle. If there is a defect in any enzyme which is part of urea cycle, so how to treat that and what can happen. Now, on the basis of the final product of oxidation of carbon skeleton of amino acids can be divided into 3 different categories. So if amino acid is giving rise to acetyl coenzyme A, and this acetyl coenzyme A can combine with oxaloacetate and enter into citric acid cycle. So in that case the amino acid is called ketogenic. The second category is the one if the oxidation of amino acid carbon skeleton is giving rise to either pyruvate or one or more of the intermediates of TCA cycle like alpha-ketoglutarate or succinyl coenzyme A or fumarate or oxaloacetate. In that case the amino acid is called glucogenic, because in these cases oxaloacetate which is produced from TCA cycle can enter into gluconeogenesis and can produce glucose. So those amino acids are called glucogenic. Then there is third category, some of the amino acids, they produce both acetyl coenzyme A as well as either of the intermediate of TCA cycle. So those amino acids are called both ketogenic as well as glucogenic. So the major difference between being ketogenic and glucogenic is that because when any intermediate, any product or any chemical enter at acetyl coenzyme in TCA cycle, it will react with one molecule of oxaloacetate and will produce one citrate. And this one citrate, after the completion of cycle, this gives rise to one molecule of oxaloacetate. So in that case, there is no net gain in concentration of oxaloacetate if acetyl coenzyme A is entering in TCA cycle. That's why these amino acids are ketogenic. They only give rise to ketone body synthesis. Whereas glucogenic amino acid, they give rise to either of these intermediates of TCA cycle, and eventually there will be gain in concentration of oxaloacetate, and this oxaloacetate can be converted into glucose. So those amino acids are called glucogenic. In fact, all the products of oxidation of carbon Skelton of amino acid, they enter into this central metabolic pathway. That's what you call TCA cycle. We have discussed about citric acid cycle in an earlier lecture, and amino acid can produce either one or more intermediates. There are 7 amino acids out of 20 naturally occurring amino acids which are converted into acetyl coenzyme A. Those are leucine, isoleucine, threonine, lysine, phenylalanine, tyrosine and tryp. In short, they are called KL50. Just to remember it, KL50. These are one-letter abbreviations of amino acids. So out of 20 amino acids, there are 7 which are converted to acetyl coenzyme A, and out of those 7, there are only 2 amino acids which are purely ketogenic. Ketogenic means they only produce acetyl coenzyme A, and those are leucine and lysine. So here K and L. So K and L are the only 2 amino acids which are pure ketogenic. They produce only acetyl coenzyme A, and rest 5, WIFTY, W-I-F-T-Y, these 5 amino acids, they produce acetyl coenzyme A along with some of the intermediate of TCA cycle. So WIFTY are both ketogenic as well as glucogenic. And rest 13 amino acid, they are purely glucogenic in nature. So that's how you can remember them. KL50 are the one which are converted into acetyl coenzyme A, and out of KL50, KL are purely ketogenic, 50 are both ketogenic and glucogenic, and rest 13 amino acid, they are purely glucogenic in nature. So out of those 13, 6 are converted to pyruvate, where like alanine, cysteine, glycine, serine, they are purely converted to pyruvate, so they are called pure glucogenic in nature, whereas threonine, tryptophan, the red color shown here, they are both ketogenic as well as glucogenic. Then you have 5 amino acids which are converted into alpha-ketoglutarate, another intermediate of citric acid cycle. Those are arginine, glutamate, glutamine, histidine and proline. And 4 amino acids are converted to succinyl coenzyme A, including 2 common, both ketogenic and glucogenic, isoleucine and threonine, along with methionine and valine. 2 amino acids, phenylalanine and tyrosine, they are converted into fumarate, but they also converted into acetyl coenzyme A. So those 2 are both ketogenic as well as glucogenic in nature. Then you have aspartate and asparagine. These 2 amino acids, they are converted to oxaloacetate, and they are purely glucogenic in nature. So first we'll talk about branch chain amino acid. So branch chain amino acids are leucine, isoleucine and valine, and they are very important for muscle maintenance because they are energy source for muscles. BCA, because we'll see later in later slides, these are oxidized only in muscles, brain and adipose. The liver don't have the enzyme which can oxidized branch chain amino acids. So in that way they provide a very good energy source for muscular activities, and they have ingredients which are building blocks for muscles, because all the three amino acids, they are essential amino acid, they are non-polar and essential amino acid. They make up to 30 to 40% of the essential amino acids in protein that build muscles. And these branch chain amino acid also support muscle condition, because they inhibit degradation of muscle protein during exercise. So muscles are not degraded when exercise is being made. In fact, BCA, these branch chain amino acids, they promote protein synthesis, the muscle protein synthesis. In fact, by one of the studies it was found out that they play a lot of role along with decreasing proteolysis in the muscles, so proteins are not degraded. They found to increase protein synthesis, so more protein, more muscle can be built in presence of these amino acids. They reduce appetite, and also they reduce adipose tissue deposition, so fat deposition also reduced by branch chain amino acids. And of course glutamine and alanine in muscles, which are a carrier of amino group, they are released from the muscles. Those increase because when you do muscular activity, those amino acids are oxidized, and glutamine and alanine, they are released in blood, which enter to liver and further catabolized. And of course they provide energy, modulate neurotransmission. They are new functions which have been observed for branch chain amino acids. They also modulate glucose metabolism, and many other effects they have on the body, which have been reported in 2018 in this research article. Now, oxidation of branch chain amino acid also occur in a similar way like any other amino acid. Only difference is that this enzyme is not present in liver. In fact, it is found in muscles, adipose, kidneys and brain, and the enzyme is called branch chain aminotransferase, BCAT. BCAT, the mechanism of action is same like other transaminases, which use PLP, pyridoxal phosphate, that is derived from pyridoxine vitamin B6 as a cofactor, and BCAT transfer amino group, the primary amino group of these amino acid, to alpha-ketoglutarate like any other amino acid. So L-glutamate is synthesized. This L-glutamate, it is converted into glutamine, and it goes to liver for urea cycle. So that is amino group is converted into urea, and the carbon skeleton is left, though that is alpha ketoisovalerate from valine. These are the keto forms, and isoleucine give rise to alpha keto beta methyl valerate. Leucine is converted into alpha keto isocaproate. Now these three keto acids which are produced from these branch chain amino acid, they are further degraded into corresponding acyl coenzyme A esters by enzyme called branch chain alpha keto acid dehydrogenase. So these branch chain alpha keto acid dehydrogenase is a complex enzyme where three enzymes are clubbed together, like we have seen in case of pyruvate dehydrogenase complex, which oxidize pyruvic acid into acetyl coenzyme A, and this similar mechanism this branch chain alpha keto acid dehydrogenase also has, where three enzymes are associated, and it catalyzes oxidative decarboxylation reaction, where coenzyme A is added to the keto acid, carbon dioxide is released, and NAD plus is reduced to NADH and H plus, and the corresponding ester of coenzyme of these three keto acids, those are synthesized. So in case of isoleucine and valine, propionyl coenzyme A is the final product of oxidation reactions in both the cases. In fact, in case of isoleucine, acetyl coenzyme A is also formed, and this propionyl coenzyme A is converted into succinyl coenzyme A, which enters into TCA cycle. But in case of leucine, the final product is acetoacetate and acetyl coenzyme A. So that's why leucine give rise to only ketone bodies, or acetyl coenzyme A is purely ketogenic amino acid, whereas valine give rise to only propionyl coenzyme A, which is converted to succinyl coenzyme A. So this is purely glucogenic amino acid, and isoleucine produce both acetyl coenzyme A as well as succinyl coenzyme A. So this is ketogenic as well as glucogenic. So these three branch chain amino acid, out of these three, leucine is ketogenic, valine is glucogenic, and isoleucine is both ketogenic as well as glucogenic. 6 amino acids are degraded to acetyl coenzyme A, like tryptophan, lysine, phenylalanine, and leucine, along with isoleucine. So they are converted into acetyl coenzyme A. So tryptophan and lysine, they share a common intermediate. Tryptophan is oxidized after 9 steps to alpha-ketoadipate. Even lysine also oxidized to alpha-ketoadipate, which is converted to acetoacetyl coenzyme A. And similarly, phenylalanine is first oxidized to tyrosine, and this tyrosine is eventually converted to acetoacetyl coenzyme A and or acetoacetate, giving rise to fumarate. So this fumarate enters into TCA cycle, and that's why phenylalanine and tyrosine, they are glucogenic also, along with ketogenic in nature. Then leucine give rise to only acetyl coenzyme A, so leucine is purely ketogenic amino acid, whereas isoleucine give rise to succinyl coenzyme A, so that is glucogenic in nature. Five amino acids: arginine, proline, histidine, glutamate, and glutamine. They are converted to alpha-ketoglutarate. Arginine is converted into ornithine by arginase, which is urea cycle enzyme. Urea is formed, and this ornithine is further converted to glutamate gamma semialdehyde by ornithine gamma aminotransferase, where one ketoglutarate is converted into glutamate, and even proline is also converted into glutamate gamma semialdehyde by the action of proline oxidase enzyme. Now this glutamate gamma semialdehyde is reduced by glutamate semialdehyde dehydrogenase into glutamate, and histidine is also finally oxidized to glutamate, and glutaminase that we have seen in the earlier lecture, glutaminase hydrolyzes glutamine into glutamate, giving rise to one molecule of ammonia, which enter into urea cycle, and this glutamate is converted into alpha-ketoglutarate by GDH. This enzyme also we have discussed in the previous lecture. Methionine, isoleucine, threonine, and valine, they are also associated to one metabolic disorder, because these 4 amino acids, methionine, isoleucine, threonine, and valine, they are converted into propionyl coenzyme A, and this propionyl coenzyme A is converted into methyl malonyl coenzyme A, and this methyl malonyl coenzyme A, by the help of enzyme methyl malonyl coenzyme A mutase, or this is also called as MCM, this enzyme convert methyl malonyl coenzyme A into succinyl coenzyme A, and this succinyl coenzyme A enter into TCA cycle to give energy or to take part in gluconeogenesis. So in case of any mutation into MCM, this methyl malonyl coenzyme A mutase enzyme, this reaction is stopped, and when this reaction is stopped, methyl malonyl coenzyme A start accumulating in the cell, which is also converted into methyl malonic acid, and this disease is called methyl malonic aciduria, MMA. In fact, this enzyme methyl malonyl coenzyme A mutase, it uses vitamin B12 derived coenzyme B12 as a cofactor. So coenzyme B12 is essential for activity of this enzyme, and this coenzyme B12 is synthesized from vitamin B. So if this enzyme is methyl malonyl coenzyme mutase is defunct, it has got mutation, it has lost its activity, or vitamin B12 is not able to convert into a coenzyme B12, or vitamin B12 is not being absorbed in the body. In all these situations, methyl malonyl coenzyme mutase activity is lost. As a result, methyl malonyl coenzyme start accumulating in the body, and that increases level of methyl malonic acid, giving rise to methyl malonic acid urea, MMA. In fact, not only these 4 amino acids, we have discussed earlier about in beta oxidation of fatty acid as well, odd chain fatty acid, they are also eventually oxidized into propionyl coenzyme. And in fact, in gut flora, those also release propionyl coenzyme, propionic acid, that is absorbed and converted into propionyl coenzyme in the body. So the only way propionyl coenzyme can be converted into some product in the body, that is by this enzyme MCM. If anyhow MCM is not working, in that case there is no other way methyl malonyl coenzyme can be converted into any other product. So it start accumulating, it is only converted into methyl malonic acid, so which increase in the body too much, and that give rise to condition called methyl malonic acid urea. That's a toxic effect of increased concentration of methyl malonic acid. Glycine can be catalyzed using 3 different pathways. In first pathway, glycine is converted into serine by enzyme called serine hydroxymethyl transferase, which transfer one hydroxymethyl group on glycine from N5-N10 methylene tetrahydrofolate. So in turn, methylene tetrahydrofolate is converted into tetrahydrofolate and serine is synthesized. Now serine hydratase hydrolyze serine to pyruvate. This pyruvic acid, it can enter to TCA cycle, or it can be used for synthesis of glucose in gluconeogenesis. In the second pathway, which is more prominent in mammals, glycine is oxidized to carbon dioxide and ammonia in presence of enzyme called glycine cleavage enzyme, or also known as glycine synthase enzyme. Glycine synthase oxidize alpha carboxyl group of glycine to carbon dioxide and alpha amino group into ammonia. And same time, one molecule of NAD plus is reduced to NADH, and one carbon atom, the alpha carbon of glycine, is transferred to tetrahydrofolate. So tetrahydrofolate is converted to methylene tetrahydrofolate. In the third pathway of glycine oxidation, D-amino acid oxidase oxidize glycine into glyoxylate, and this glyoxylate is further oxidized to oxalate, where NAD plus is reduced to NADH. This reaction is found in kidneys, and it's a minor pathway where glycine is converted into oxalate. In fact, in high concentration, oxalate crystallizes, and that's responsible for most of the kidney stones in the kidney tubules. Asparagine and aspartate both are purely glucogenic amino acids, and they are converted to oxaloacetate. Asparagine is hydrolyzed by asparaginase into aspartate, and ammonia is released. This ammonia enters into urea cycle, whereas aspartate is further converted into oxaloacetate by aspartate amino transferase or aspartate transaminase enzyme. That's a PLP dependent enzyme which transfer this alpha amino group of aspartate to alpha-ketoglutarate, and glutamate is synthesized. This glutamate is converted into glutamine, and glutamine that goes to liver to participate into, to release ammonia, which participate into urea cycle, and the end product is oxaloacetate. This oxaloacetate enter into TCA cycle, or it can be used for glucose synthesis in gluconeogenesis. Oxidation of aromatic amino acids, phenylalanine or tyrosine, can lead to genetic disorders. So phenylalanine is first converted into tyrosine with the help of enzyme phenylalanine hydroxylase. This enzyme use oxygen NADH molecule tetrahydrobiopterin as a cofactor. In result, water is released and tyrosine is synthesized. Any genetic defect into phenylalanine hydroxylase leads to accumulation of phenylalanine, and that state is called phenylketonuria. There's a genetic disorder that's found in people who have a genetic defect, a genetic mutation into phenylalanine hydroxylase. Such people, they cannot digest phenylalanine, so that start accumulating in the body. Then this tyrosine is converted into para-hydroxyphenyl pyruvate with the help of enzyme tyrosine aminotransferase, where amino group is transferred on alpha-ketoglutarate, which is converted into glutamate. So this glutamate is converted into glutamine, and that's how that goes to liver and participate into urea cycle. If there is genetic defect into tyrosine aminotransferase, the disease is called tyrosinemia, where tyrosine concentration in the body, it accumulates, it increases. Now this para-hydroxyphenyl pyruvate is oxidized to homogenticate by enzyme para-hydroxyphenyl pyruvate dioxygenase. Genetic defect into this enzyme para-hydroxyphenyl pyruvate dioxygenase leads to a disease called tyrosinemia 3, where para-hydroxyphenyl pyruvate increases in concentration. Now this homogenticate is further oxidized into mallyl acetoacetate by enzyme homogenticate 1,2-dioxygenase, and in fact genetic defects have been observed into this enzyme as well, where homogenticate concentration is increased in blood and urine, and that state is called alkaptonuria. After this, mallyl acetoacetate is isomerized into fumaryl acetoacetate by mallyl acetoacetate isomerase, and fumaryl acetoacetate is hydrolyzed into fumarate and acetoacetate by enzyme fumaryl acetoacetate. Defect into activity of fumaryl acetoacetate leads to accumulation of fumaryl acetoacetate in the blood and urine, that is called tyrosinemia 1. And further this fumarate, this enter into TCA cycle, whereas acetoacetate is converted to acetoacetyl coenzyme A, and then finally acetyl coenzyme A. So that's how phenylalanine and tyrosine, they both are ketogenic as well as glucogenic amino acid, because fumarate is glucogenic and acetyl coenzyme A or acetoacetate, that's a ketogenic in nature. In fact, in case of phenylketonuria, cells acquire a different pathway for oxidation of phenylalanine. So phenylalanine is first converted into phenyl pyruvate by aminotransferase, where pyruvate is converted into alanine by this PLP dependent enzyme, and this phenyl pyruvate is converted into phenyl acetate and phenyl lactate. That's why the patient having phenylketonuria, they have very high concentration of phenyl pyruvate, phenyl lactate and phenyl acetate in the body fluids. And all these genetic disorders, they can be only controlled by limiting diet. For example, in case of phenylketonuria, the phenylalanine concentration in the diet is limited to the minimum possible, so that excess phenylalanine is not available for oxidation, so that disease symptoms can be taken care of. In fact, tryptophan has been used into the intermediate of tryptophan oxidation. They are used for synthetic reactions also, like indolling of tryptophan that give rise to synthesis of nicotinate niacin, which is used for synthesis of NAD plus and NADP plus cofactors. Similarly, indolling is also converted into indole acetic acid in plants, which work as a plant growth hormone, and tryptophan is also converted into serotonin, a neurotransmitter. Methionine is also converted into a very important cofactor, S-adenosyl methionine, SAM, and this SAM is along with the tetrahydrofolate. SAM also works as a very good transporter of one carbon atom into different carbon transferring reactions. So this synthesis of SAM from methionine is called activated methyl cycle. So methionine react with ATP in presence of enzyme methionine adenosyl transferase, and it gives rise to synthesis of S-adenosyl methionine, SAM. Now this SAM is capable, because you have methyl group here with sulfur, so it's capable of transferring a methyl group to different substrates. In fact, this is a known to be 1000 times better potential of transferring methyl group in comparison to tetrahydrofolate because of presence of this sulfur atom here. And this SAM is converted into S-adenosyl homocysteine by a variety of methyl transferase enzymes. So in that case, this methyl group is transferred into some substrate, and what is left is S-adenosyl homocysteine. This S-adenosyl homocysteine is hydrolyzed, so adenosine is released and homocysteine is synthesized. Now this homocysteine, one methyl group is transferred on the homocysteine from methylene tetrahydrofolate by enzyme methionine synthase, and methylene tetrahydrofolate is converted into tetrahydrofolate and methionine is synthesized. Methionine synthase use coenzyme B12 as a cofactor for this methyl transferring reaction. Several defects into enzymes of urea cycle have been observed, because most of the reactions of urea cycle are irreversible in nature. So in that case, it's very easy to know which enzyme is defunct in the urea in case of any defect. For example, if citrulline is found in high concentration and urea cycle is not occurring, it simply means arginosuccinate synthetase ASS enzyme is defective, because this reaction is also irreversible in nature. So that's why by measuring which intermediate of urea cycle is present in high concentration, it can be found out which enzyme has got mutation or which enzyme activity is lost. In that case, hyperammonemia occurs, because urea cycle cannot succeed if there is any defect into the enzymes of urea cycle. So in that case, ammonia concentration rise in the blood and leads to hyperammonemia. So this hyperammonemia is treated by giving two compounds, benzoate and phenylbutyrate. So basically, objective to give this compound is to reduce level of ammonia in the body. So benzoate is converted into benzoyl coenzyme A. It uses ATP in the reaction, and this benzoyl coenzyme A, it react with glycine and form benzoyl glycine. That's also called hippurate. So this hippurate is non-toxic in nature. It is secreted out from the body in urine, and it uses glycine here. So glycine basically synthesized from glutamine. So glutamine is used to synthesize glycine, and we know from the previous lecture, glutamine is basically carrier of ammonia. So in turn, ammonia concentration is regulated in the body, because to synthesize more glycine, you require ammonia to be used in form of glutamine. And in the second molecule, phenylbutyrate, this drug is converted into phenyl acetate by beta oxidation reactions, and phenyl acetate is converted into phenyl acetyl coenzyme A, that also use one ATP molecule. And now this phenyl acetyl coenzyme A, it react with glutamine directly. So, and it forms phenyl acetyl glutamine, and this phenyl acetyl glutamine, this is also secreted out of the body in urine. So that's how glutamine concentration is controlled by these compounds. And because glutamine comes from ammonia, so in turn ammonia level is maintained by phenyl butyrate and benzoate compounds, which are given to patient who have defect in the urea cycle enzymes.